Hello, OIS Podcast audience. This is Rob Rothman. Happy to be speaking with you again today. By way of a brief introduction, for those of you who don't know me, I am a practicing glaucoma specialist in New York. I spend about 50% of my time in active patient care, and the remainder of my time I spend running InFocus Capital Partners, which is an ophthalmology-specific venture capital fund. We have closed our first fund, currently navigating our 12 portfolio assets, and we have just begun active capital raise for Fund 2, which we hope to reach first close sometime mid to late Q1 of this year. It is a distinct privilege today to be speaking to Prasad Sunkara, who is the CEO of an incredibly interesting company that we're going to spend some time talking about called Eyegenex. And not to be confused, for those of you who are listening, it is Eyegenex, Eyegenex. There are other Eyegenexes out there that have different spellings, but this is Eyegenex. He is also the managing director of Seed to Fruit Fund, which we'll spend a little time talking about as well. And Prasad is probably the best example that I can think of a modern Renaissance man. And once we go into his background of all the things that he's done over the course of his career that have led to where he is today, I think everybody will be thoroughly impressed. Prasad, thank you so much for taking the time to speak with us today, and welcome to the podcast.
It's a great pleasure, Rob, to talk to you and share some of the information about myself and also about Eyegenex.
So I had the opportunity to meet you very briefly at the OIS meeting in San Diego. And, as a glaucoma specialist, I have a particular interest in what you're doing. And we'll talk a little bit about glaucoma, the management of glaucoma, and we'll talk a little bit about why what you're seeking to do is important for the future of glaucoma care. But I think it's really important for people to understand why this is exciting. I think it's important for them to understand a little bit about your background. So can you just tell us, sort of go through a little bit of history of your very long and successful career, both in corporate finance, research development, management, multiple disease states. How did it all start? Where did you come from, and kind of how did you end up at Seed to Fruit and Eyegenex?
Yeah, it's a honor and pleasure to share some of that information. I'm originally from India, as you can guess, from a small coastal town in the southern part of India called Machilipatnam. That means fish town, literally. I came from a farmer's family, but it so happened that my father was the first one to go to university and became a banker. But in a lot of aspects, he was an entrepreneur too. It was, he actually a self-made man. And then that had a lot of influence in me. And when I finished my high school, I ended up going to agricultural sciences, did a BS in agriculture. But I was influenced tremendously by the work at that time in the molecular biology, Watson and Corona and all this. So I actually left agriculture and did my master's in microbiology and biochemistry. And then I was fortunate to go into get admission at one of the leading institutions in India called Indian Institute of Science in Bangalore. I did a PhD there. And I did very well and gotten some award from the Prime Minister of India and all that. But what is more interesting is that I got fellowship at the MD Anderson Cancer Center to work in cell biology. So I had an opportunity to do also in tumor immunology at NCI, but I felt I'm not really have not such a great background in immunology. So I ended up, I wanted to look at the up and coming cell biology, and I joined MD Anderson Cancer Center. And after a couple of years of postdoc, they offered me an assistant professor job. And I was there for 4 more years and was successful in getting 2 R01 grants. And I was doing extremely well. Then I was recruited into a pharmaceutical company at the time called Marion Merrill Dow in Cincinnati, Ohio. They were trying to start a tumor biology group, and then they recruited me to come and be head of that. Eventually, it became a department, and so I was there for 13 years, worked in a number of areas, number of enzyme targeted inhibitors, catalytic inhibitors, and it was a very interesting time in my life and career. I was involved not only in the drug discovery, but also preclinical development and part of clinical development as the lead for the oncology and antiviral. At the time, HIV is one of the active areas. So, but then after a while, I wanted to go more into preclinical and clinical aspects of drug development. And it's very hard in a big pharma to do that move because everybody says, oh, you are a discovery guy, So I actually left Marion Merrill Dow after 13 years, came to San Diego. I joined, a startup, became a product development guy, director of product development, and learned the virtual development. And I was there for 4 years with a company called Neurocrine Biosciences, and that company still exists and doing very well. But then I got an opportunity to go back into cancer. The other one was a CNS company. So I moved as a VP of another cancer biology company. The 90s were a heyday for biotech. The valuations were not that great, but it's easier to get funding and get going. And if you do reasonably well in 2-3 years, there was an exit, a merger mostly, or maybe sometimes possibly IPO. So that company was taken over in a couple of years, and then I joined another company and a higher executive VP of R&D, and that we exited in a couple of years. So when the last company got acquired in 2000, I wanted to take a break and see what I want to do. And then fortunately, I met with a couple of professors at the University of Michigan medical school, and they had some funding from state of Michigan. Those days they had money, I suppose. But they didn't know how to go about it. They're good in writing grants, So I met with them, and we formed a partnership and started a company. We leveraged the funding, and we started a company called Molecular Therapeutics. The idea of molecular therapeutics had transformed into a holding company where molecular therapeutics focuses on drug development. And then we started a CRO, kind of preclinical CRO called Molecular Imaging Research. These two guys were directors of University of Michigan Molecular Imaging Center. So essentially we did acquire, animal MRI, PET, and bioluminescence imaging, all these things, and we were focusing on cancer efficacy studies. So essentially the same animal we can follow through instead of sacrificing them. It was a very interesting, and we'll do it as for close to as GLP so that they can, submit all that data to FDA. So we worked with a number of biotech companies, some pharma, mostly biotech. And as a result, we did quite well. We grew that to 50 people. And eventually, both the companies were acquired by Charles River Labs. As you know, it's one of the big, CROs in Boston, near Waltham. And then I stayed back in Ann Arbor, mostly because my son was doing a residency in Pennsylvania at that time. So my wife didn't want to get back to California. Our intent was always to come back to California. So we stayed back. Then I became CEO of 2 other companies, helped them to grow. And eventually when the last one was acquired, we moved back in 2015 to San Diego. And I got introduced to a very active angel group called at that time called TCA, Track Coast Angels in San Diego. And I got involved with them, and I invested. I was part of the life science screening committee, and as with time, I became the chair for life sciences, and was the chair for about 4.5 years, spending a lot of time, but then what I really enjoy is the learning process of the new technologies when working with entrepreneurs, because I'm passionate about startups. So talking to these people, trying to help them, and going through the whole DD process. But one of the other positives was I made friends, and with a lot of those members, and I still have a lot of camaraderie and all that we developed. And so when I stepped down, I wanted to go back to what I was doing before, as even when I was in San Diego and Ann Arbor, was I was quite successful in licensing university technologies, patents, working with the professors, like I did with the Michigan folks. And I worked with Wayne State, I worked with Stanford, I worked with even small university, the Southern Illinois University, and Wisconsin and Oregon. So, working with them, licensing technologies and starting a company and trying to, fund them and build a team, take it forward. That's what I was doing. So I was successful in about 5 of the 6. I was able to do that. And so I wanted to get back to that model again. So I formed a family fund and also a foundation studio and an accelerator called Seed to Fruit. That was in 2021. And I was actually 22. I was actively working. Once I stepped down, I was spending a lot of time trying to find technologies. I must have looked at about 800 technologies from different universities all over the country and selected 40. I had a group of advisors, within this new fund group, and we pared down to about 40, and then to 10, and then top 3. So the first company we started was Eyegenex of that. And then the second company is one that I have a CEO. We just got funded, is Paroral Biosciences. The model is you license the technology, have the professor actively involved in the company, give equity, small equity to the university, and significant equity to the professor, maybe some of the collaborators and some of the advisors and board members, and majority of the equity would be with the Seed to Fruit until we start raising funding. So we raised about $750,000 for the Eyegenex to do some of the extension studies that we wanted to do. We planned, and especially non-human primates, we wanted to beef up the data we had in mice and rats into non-human primates, which we completed one study. We're in the process of initiating a second study. So, we think we have. I'll talk to you more about Eyegenex when it comes. Right. But that was my path, and I'm a continuous learner. I'm so excited about and passionate about the startups, and I'm always looking. But my intent is to start a company, be an interim CEO, bring it to certain stage, and pass on the baton, because it's always good to have some equity, cash to give to a good CEO rather than equity. So that's why that's what I plan to do in future.
Well, let me tell you, I think Craig and the team at OIS will attest to the fact that that is the longest amount of time that I've ever kept quiet during somebody's introduction of themselves. So there's no question that is a fascinating, story of growth into this space. And when I think back to all the things that you've just said, I mean, you basically have, you started out in basic science research with, a passion towards life science to some degree, and ended up with not only science experience, but grant writing experience, corporate development experience, business development experience, venture experience, startup experience. I mean, it's like the entire sort of universe of life science development sort of exists inside of your pathway, which is kind of unique. And I think that's maybe the reason that I use the term Renaissance. Most of the people that I think we meet who end up as CEOs of companies have been corporate people who have developed skill sets inside of corporate, but to have, your background in all aspects of development is definitely unique. A couple of questions. Seed to Fruit, just to clarify, is that a funded enterprise? Do you have capital that's been committed by investors that you plan to deploy, or do you identify the asset from the university and then go raise capital for them?
Yeah, it's not funded. It's just me. It's my, it's completely a LLC, rather a family fund.
Right.
So, but I go in and find and
You'll raise
and
money. So you'll raise money for the
well,
effectively for the seed round or the friends and family round, let's call it. You'll
yes,
raise the capital to sort of identify. And are the companies that are selected not just from ophthalmology, from all disciplines of
no,
healthcare, right? All
no,
all life sciences.
right. Just to clarify, though, I go and find the companies. I invest whatever time and money I need to license that patents into the company. I form
Right.
I incorporate a Delaware corporation, bring it in. At that point, I go out and raise seed funding from New Fund Ventures. That's the new name for our TCA that I used
Right.
to lead in the life sciences,
Right.
and then family and friends. So at that point, I try and bring a CEO or other colleagues that are part of the new fund to be, because there are a lot of, like Steve, you probably uh, don't know him that well, but you met with him.
Right.
He was a retired emergency doc, and he's so passionate. I mean, all these people are so
Right.
excited about startups. So I bring them in, and then we do whatever milestones to add value. And once we do that, then we go out and raise the next round from people like InFocus, Excite,
Right. Right.
whatever.
So,
Yeah.
the reason I'm asking is because there are lots of people who listen to these podcasts, and the question is, are you interested in having entities that may not be university based, but are at the, let's say, seed stage present to you? Are you looking for access to investable opportunities at the seed stage, or are you intent on continuing to go out and seek the things that you find interesting from academic institutions? Because it's important for the audience, I think, to know if there's an opportunity for them to contact you and present where they're at.
Yeah, I would prefer, I would say 80% from I do my own from the universities. Like you mentioned, I was an academic at one point. I'm still a scientist. I enjoy science. I can understand science. So it's easy for me to communicate and discuss with the professors.
Right.
And so that's what I really enjoy, and I like to bring in. And another reason is by the time I want to give my investors an ability to hopefully a good return. So this way, the dilution is less.
Right.
So I don't have to share significant equity with already a seed company, But sometimes a professor has already a company he wants me to have. And at that point, it's not that bad, because one professor, he probably wants to go back to being a professor. So we give decent equity, and he will be a consultant or chief scientific advisor. And I would be, but like I said, 80% my own, 20% seed. And then to answer one of your other questions, I'm agnostic to the field, although I did a lot of work in cancer, medical devices, but this is my first ophthalmology company.
Right.
I'm really interested. When I saw this, I was so excited because I have a personal interest in glaucoma because you probably know a lot more than I do, Rob, is in Asia, especially in Japan, China, and India, there's a lot of this NTG, normal tension glaucoma, and in India, I can say my brother-in-law, my wife's brother, lost one eye because they don't go on a regular examination to an ophthalmologist, and then their second eye was half gone, the sight, before he started on these drops. And then as a result, we started going to ophthalmologists, me and my wife. We found out we had a little increase in our eye pressure too, IOP. So we started taking drops. And obviously, when we had the recently cataracts, they put in a shunt, so we don't use them. So that's another motivation. I wanted to see, is there something for the treatment of glaucoma rather than prevention? Because as you know, 10 to 15% of these primary open angle glaucoma end up progressing into glaucoma. So is there a way that we can intervene? That's the other motivation. When I saw this, I was so excited. It so happened that Professor Nagaraj at University of Colorado is also a very well respected ophthalmology scientist, in this field. So, and he's an integral part of our company. I mean,
yeah. Well,
amazing.
Look, we're motivated. We're aligned in that regard, and I've said this on multiple podcasts before, that one of the reasons that In Focus came to be, I have a long history of investment activities in venture investing, but being an ophthalmologist and having some success in funding deals in ophthalmology, one of my goals is to be able to, treat glaucoma from the golf course. It's one of my personal, I feel kind of like if there's a way for me to get all the information I need without actually being in an office, it's going to be great. Not only, as a joke, because it's funny to say that, but when you think about all the people in the world who don't have access to an actual practitioner somewhere who lose vision to glaucoma, that's really the extension of that, is if I can treat it from the golf course, then people who are in, rural areas of countries that, may not have as many, practicing physicians to manage the disease, then you're really going to save a lot of blindness. So we're aligned in that regard. You unfortunately have to deal with it as a patient, but I think the motivation is the same. Let's talk about Eyegenex, because I understand now, it was, one of the future questions was why, after looking at all of the things that you've looked at over the course of your evaluative process, did you end up with a glaucoma company? So you kind of have answered all, that already. And knowing that you've seen so many deals and so many technologies, what is it about Eyegenex that you want to share, and why do you think it's a unique opportunity?
Yeah, well, I think I would say three things. One is my personal experience, family, and me and my wife. And then second is, obviously I didn't go out to look for glaucoma technology rather, but it so happened when I saw this, and I actually did quite a bit of due diligence working with ophthalmologists, retinal surgeons, and my own advisors, and but then as you know, the endpoints are still difficult, and so I was discouraged essentially to go forward because of these endpoints. There are no approved endpoints, and still the only IOP lowering. That's what people are looking at. But I felt strongly that as we go along, develop science and find markers, and in a couple of years or so, when you go and do the clinical trials, there may be an advancement. And as you can see, this Francisco Caldera, she's working on a way to look at apoptosis in patients. And then Franz Gruz, I think, in Germany, is also looking at some glaucoma, testing using some biomarkers. So things will improve. That was my belief. So and then I was so the second one is I was so impressed by what Professor Nagaraj has done, and he's is full of ideas. We work together it turns out we work together very well. We have a small team, but very good team. And so those are the few things that really impressed me with this area and the technology. We not only have, and the other thing is obviously the gene therapy, and we not only have a gene therapy, but also a peptide therapy asset that we are developing, and we have a lot of animal data and very, very effective in these animals. Obviously, our initial monkey data suggests that also. So we're pretty excited about the next couple of years, and in the meantime, we got to raise our second round and get going on the
Of
enabling.
course. Well,
Yeah.
I think it's interesting. I agree with you that for glaucoma in general, it's really been about IOP reduction, which is really not what we need in terms of finding let's say long-term or treatments for glaucoma, right? We know that IOP reduction is important, and there's lots of companies developing surgical products, and there are some new chemical entities that will be coming out that may help reduce IOP, and we know that that's an important part of the process. But for in focus specifically, we've looked at the neuroprotection as the real sort of, even if it's an adjunctive therapy, but a real way to try and prevent cellular death. And there have been some things that are promising in terms of clinical trial design that I think will benefit both of our assets and your company, in terms of how the FDA will allow for a company to prove that it may be neuroprotective using surrogate visual field data, right? And we
Yeah.
have novel technologies, like dark, right, which may
Yeah.
become an important part of how we evaluate. A disclaimer: we are not an investor in the company that is involved with dark, but I think it's an important technology that may help give us some actual cellular data regarding ganglion cell function in glaucoma, cell death, down the road. And like you said, there are interesting biomarkers coming around. And I think that you're right. I think a lot of people got very, very scared from some of the failures in the past with drugs that were looking at, let's say, neuroprotection as treatment for glaucoma, and I think that will change. So I agree with you that makes the glaucoma therapeutic space a lot more investable and a lot more cost effective when it comes to conducting clinical trials. I don't know exactly what the Eyegenex technologies do. I don't know how much you want to share about that, and where you are in the clinical development. Probably not a whole heck of a lot. But ultimately, I think that there is a real sort of developing simplistic or simpler approach to getting things approved.
Yeah, I would be happy to share if you have time.
Sure, go ahead. Let us know.
Yeah.
Let's hear what you
Yeah.
guys are doing.
So we have two assets. One is a gene therapy. We're focusing on small heat shock proteins as an anti-apoptotic protein, and a peptide, about 25 amino acid peptide from the same protein, 19 amino acids plus a cell-penetrating protein attached to it. So we have extensive data using microbeads, surgical laser-induced IOP, and a number of other models to show that these products are quite effective in mice and rat models. But what is really interesting is one of the studies we did with the gene therapy product. It's an AAV2 with a promoter specifically expressed only in retinal ganglion cells, so it doesn't go to any other cell. Professor Nagaraj, we call him Ram, his first name, Ram, actually did this study wherein he had three groups of animals. One is naive animals, untreated. The other one is a glaucoma animal, induced glaucoma with microbeads, and then a vehicle-treated group, second. The third one is glaucoma plus the gene product intravitreally injected. And then he looked at them after a year. And what is amazing is this gene is expressing the protein even after a year, only in retinal ganglion cells, significant expression. And as a result of that, the retinal ganglion cell counts, the death is so mitigated, is equivalent to the naive animal cell count. In fact, it'll more. Probably the naive animals with age lost some RGCs, whereas here they're being, prevented from cell death. And where compared to there is about 20% reduction in the RGC cells in the vehicle treated. Okay, so
and that's intravitreal injection, single intravitreal injection.
Yes, single intravitreal injection after a year. But when you look at it's a preliminary data, fewer animals. When you look at the vision by PERG, what we found is that naive animals have let's say go back to the vehicle treated animals, they're almost blind compared to the naive animals, whereas the gene therapy treated animals were as good as the naive animals. So the animals numbers are small, but it's an indication. Preliminarily, there is I think we're so excited to see that data, and
that's exciting. I mean, that's true neuroprotection.
Right, and I presented that briefly at OIS, but just to go back and as an extension, we wanted to see, well, obviously mice are not humans. So we wanted to go and look at, in monkeys, the optic nerve is similar to humans. So we actually were able to do a small study. Initially, we did this with a company called Versio. They use African green monkeys and Saint Kitts. So we worked with them to see whether we see expression of this gene therapy product in retinal ganglion cells in monkeys. And we did the same thing. We did a dose response, low, medium, and high dose, and looked for expression after a month. And we just got the data back in about a couple of weeks ago, and it was beautiful. We could see a dose-dependent expression of this gene, the gene product, which is this protein, not only in soma, but also in axons. So it's really beautiful.
Fantastic.
Right. And I was told that the RGC cell died from axon. So we have this expression throughout, RGC cells. So what we're trying to do is we're going to do, these are not publishable, but we de-risk as much as possible so that when we go to the investors, say, look, this is not something we go out and publish, but at least we have this data in small number of animals. So we're actually going to do a glaucoma model in these monkeys, a small study, because we know the dose already where there's a good expression. We use the one single dose and use a naive and a glaucoma, placebo, and the high dose, what we used in the last study. So we should have that data in 3 months, and we are planning before that filing for a pre-IND submission and hopefully hearing from FDA, and then having this data. With all that, we want to go out and meet folks like you and others and trying to raise about maybe $6 to $8 million. We want focus on the gene therapy, but there, maybe we don't want to dilute our investors tremendously. So we might go and raise more later for the peptide. We are working on a slow release formulation of the peptide, also using some of the funds.
Yeah. So what's the peptide? So let's just spend 2 minutes. What is the peptide therapeutic? What is that?
Yeah, peptide therapeutics, like I said, is a 19 amino acid segment of the same small heat shock protein B1, which has retained the activity. And we added a cell-penetrating peptides, which is about 6 amino acids to that. And then it actually tremendously increases the activity and the uptake of the peptide. We have a lot more data, in fact, from the peptide, and we were thinking of using it for acute angle glaucoma, and in combination, like you mentioned, adjunct to IOP lowering agents, because but the number of patients are not that significant, but that may be something that we could just inject when somebody comes to ER, maybe an ophthalmologist can come in
Right,
and
to protect the optic
yeah,
nerve from an acute
right,
situation, right?
right. That's what we were thinking of. But what is not, not published or patented yet is there seems to be some other activities um, these peptides have in the ophthalmology space, which we want to pursue too. So as soon as we file patents, we would be able to, share that with the next round of investors.
Well, Prasad, I have to tell you, it's pretty exciting to hear what you're doing. And it's nice for us to start to hear that there is actually some, you are one of a handful of companies that has, come out as pursuing gene therapy for glaucoma. And it's exciting to see. I think the whole gene therapy space in ophthalmology has been taken over by inherited retinal diseases for the most part. And it's nice to kind of see that people are realizing that glaucoma is also a target for gene therapy. We think gene therapy is going to be an important part of the treatment algorithm for patients with multiple different ophthalmic diseases in the future. And hearing what you're doing sort of gives me some encouragement that that's going to hopefully continue to propagate in the world of science as we move forward. I'm excited personally to interact with you as an investor over the next few months. And we certainly will look forward to seeing all the information you have to present at that time. But until then, I think on behalf of the entire OIS audience, I want to thank you for an incredibly illuminating discussion. I think your background is pretty unbelievable, and I'm glad to hear that you've decided to commit some of your talents to ophthalmology. Hopefully, I think maybe together we can figure out how to cure glaucoma one day.
Thank you very much. I would love to work with you, Rob. And obviously, I'm not an ophthalmology vision scientist, but I think I can bring a lot of things to the table, and hopefully bring other ophthalmology folks into the company as we move along.
Yep. Well, listen, thanks for your time today again to the OIS podcast audience. Eyegenex, Eyegenex. Take a look at them. Exciting stuff going on there. And certainly with Prasad Sunkara at the helm, I think they have a good chance of being a successful opportunity, hopefully something that will help patients down the road. I look forward to speaking with everyone again in the future. And thanks again for listening to the OIS podcast.